Hello, ladies and gentlemen. I hope you are doing well and that you are able to find some rest during this kind of crazy time. Thank you to those of you who have been communicating with me. Thank you to those of you who have been reaching out with questions, comments, or concerns. It's really helpful. It helps me do my job better for you. So continue to do that the next kind of couple days that we have left of this, hopefully. And thank you to those of you who are finishing those discussion questions daily. Make sure that they are done by 3 p.m. every single day. That gets you your attendance for that day. If you do not answer them, you will be marked absent. Also, please do not write present for your response to them. You will be marked absent as well. Okay, so today we're talking about incomplete dominance and codominance. This is something that kind of goes against everything that we've talked about up until this point. So I highly encourage you to kind of stick with it. You might actually find that it's a pretty simple concept. And as always, there's additional resources and links in the description of this assignment to kind of help you understand this. But let's get started. A lot of the vocabulary that we're going to be using in today's lesson is review. It's stuff that we've already heard before. It's stuff that we've been using before. So none of this should be brand new information except for the two at the bottom. Again, a dominant allele is one whose trait is always going to show up in the organism if one of the two alleles is dominant. In other words, if one of the two alleles that is there is a capital letter, that trait will always show up. An allele is just a different form of a gene. Heterozygous individuals are organisms that have two different alleles for a trait. So often our heterozygous alleles or genotype look like capital and then lowercase for whatever letter that you are using in any kind of circumstance. So that's what a heterozygous individual is. And these guys are going to be really, really important in today's lesson. That is because we're talking about incomplete dominance. So one allele is not completely dominant over the other. Rather, the offspring is a blend of the two. And we're talking about codominance, which is a condition where both alleles are fully expressed independently. So both are fully expressed separate of one another. Now, let's kind of get into what this actually means. Before we do that, I want to take a minute to just review some basic concepts, reinforce them. That way, incomplete dominance and codominance makes sense. Up until this point, we have been talking about Gregor Mendel's interpretation of genetics, what otherwise known as Mendelian genetics. And when Mendel was doing his experiments, he said that there were dominant alleles and recessive alleles. Based on his information, those dominant alleles that he had more frequently showed up because if there is a genotype with a dominant allele or a capital letter, it always was expressed. So in his experiments, those genotypes gave purple flowers. Hence why he called them dominant alleles. It showed up. It was more often or more likely for him to get purple flowers. He called recessive alleles recessive because they were less frequent or they were less likely to show up in his experiments. And he realized that recessive alleles only show up if both alleles happen to be recessive. So when we have a homozygous recessive genotype, we usually got in his experiments white flowers. Now, Gregor Mendel's interpretation of genetics is really, really simple and very mundane, whereas today we're going to talk about some really cool things that we've discovered that our genetics do. All right, let's briefly review heterozygous versus homozygous. The prefix hetero means different. So in a genotype that is heterozygous, you are going to have two different alleles, capital A, lowercase a. Now, the phenotype that is expressed will always be that dominant trait, and that has been proven by Gregor Mendel's law of dominance, going back to our basics of genetics video. Homozygous genotypes have two alleles that are the same. So big A, or little a, little a. A homozygous dominant individual will show the dominant trait. A homozygous recessive individual will show the recessive trait. So based on Gregor Mendel's interpretation of heterozygous and homozygous, he said that if we have an individual who is big B, little b for eye color, and let's say that big B is brown and little b is blue, based on the law of dominance, we are going to have brown eyes. Now, for homozygous individuals, if I have two capital letters or a homozygous dominant genotype, that individual will have brown eyes. For my homozygous recessive individuals who have two recessive traits in their genotype, they will show blue eyes, or that recessive trait. Therefore, if an individual is homozygous dominant, they will express the dominant trait because they have the dominant trait for both of their alleles. If an individual is heterozygous, their phenotype will also express the dominant trait. And if an individual is homozygous recessive, their phenotype will express the recessive trait, right? So if I have homozygous dominant, big B, they're going to have brown eyes. If capital B, for our dominant trait, is brown eyes. If I have an individual who is homozygous recessive, or little b, and little b is the recessive trait of blue eyes, that individual will have blue eyes. If my individual is heterozygous, meaning big B, little b, they will still show brown eyes because of the law of dominance. Now, let's briefly review that. Again, Mendelian genetics is very, very simple. So when Gregor Mendel was doing his experiments, he had a very simple understanding of what was happening. It was really important. Don't get me wrong, I'm not downplaying his discovery. He gave way for all of the stuff that we're going to talk about today. But there are exceptions to this rule, right? The law of dominance says that wherever or whenever there is a dominant allele expressed, that dominant allele will always show up in the phenotype. Therefore, this bullet point right here, therefore, in heterozygous, so individuals that have two different alleles, in a heterozygous individual, dominant alleles will always be the phenotype. Even though there's a little recessive letter B here, we will still always only show brown eyes. However, there are exceptions to this rule, and there are two exceptions: incomplete dominance and codominance. Now, these things are really, really cool, and we're going to talk about why they happen, but just know that from this point forward, we are only talking about heterozygous individuals. We are no longer talking about homozygous. So we are only talking about individuals that have two different alleles in their genotype. That's it. We are no longer talking about anything else in this lesson. Let's get started. Sometimes in heterozygous individuals, we have this thing called incomplete dominance happen. This is when a dominant trait and a recessive trait blend together to make some sort of middle phenotype or intermediate phenotype, some kind of blend of the two. Now, flowers most often portray this and most regularly do, so we're going to use them as an example. And a snapdragon is a type of flower. It's the flower that you see to the right of the screen. So let's look at our example. When a red snapdragon breeds with a white snapdragon, we end up with a possibility of having pink offspring. So incomplete dominance is when we have the dominant trait plus the recessive trait producing an offspring that is kind of in between. Think about this. It's like taking paint. When you were a kid, you were given the chance to kind of blend different paints together, right? You put yellow and blue together to make green, and it's the same thing here. If you have red paint and some white paint and you mix those together, you are going to end up with pink paint at the end. So incomplete dominance is almost like taking paint and mixing it together. There's the dominant trait and the recessive trait, and because they are both there, both in the genotype here, we're going to end up with something that's in between. Now let's talk about how this could happen. So in the previous slide, we saw that there was a red flower and we want to mix that or breed that with a white flower to see what kind of offspring we are going to get. And we know for a fact that both of these flowers can show incomplete dominance or this blend of traits. So I'm going to try and figure out what my chances are of having pink flowers, because again, red plus pink or red plus white yields pink. Think of it as paint. Red paint, mix it with some white paint, you get pink paint. So let me briefly do my Punnett square here. I drop down all of my alleles from the top. I move all of my alleles to the side, and I can see, based on my Punnett square, that I have a 100% chance of having pink flowers as my phenotype. So when we talk about incomplete dominance, again, we are looking at heterozygous individuals, offspring that have two different alleles in their genotype. Those are the only ones that we care about because those are the only ones that can show incomplete dominance. If I have offspring that have big R, they're just going to be red. There's no chance of them showing that blend because I don't have two different alleles. And it's the same. If I have offspring that are little r, they're just going to be white because there's no chance of them having a red allele in there. So anytime we talk about incomplete dominance or the chance of having pink flowers, we are looking at heterozygous individuals or those who have two different alleles in their genotype. So to wrap up this idea of incomplete dominance, we need to know that a incomplete dominance is a blend of dominant and recessive alleles. It's the dominant and the recessive alleles acting like paint and coming up with some sort of intermediate or middle phenotype. I have a red flower, I have a white flower, I breed them together, I can end up with pink flowers at the end. Now, we can see incomplete dominance in humans, meaning my dominant and my recessive alleles working together to create this kind of blend. Now, we most often see this with hair type. If you have one parent that has straight hair and the other parent has curly hair, you are often likely to have wavy hair if you have a heterozygous genotype or two different alleles in your genotype. We can also see this with skin color. If your parents happen to have two different color skin colors, not only could you be closer to your mom than your dad, you could also have kind of a perfect blend between the two. And we can also see this in eye color. How many of you have seen a person that has, and I'm going to draw kind of a creepy eye, sorry, it's just art ability. How many of you have seen a person or you have blue eyes and then on these blue eyes, I don't have brown as an option, but you have a little bit of brown around your pupil. This explanation or this shows that we have some sort of incomplete dominance happening in eye color. And we're going to talk about more specifically how this happens because incomplete dominance doesn't follow Mendel's law of dominance. Incomplete dominance says that we can have a blend of both a dominant and a recessive trait instead of just the dominant phenotype showing through. Now, the second exception to Mendel's law of dominance is this thing called codominance. This is when we don't really have a recessive allele. It doesn't really exist. Rather, both alleles in the genotype are expressed or shown at the same time. Neither of them get masked, however, and they don't even blend. We can see both of them independently on their own. This can only happen in heterozygous individuals, those who have two different alleles in their genotype. We cannot see this with individuals who are homozygous or have two of the same letters in their genotype. All right, so let's take a look at the picture on the right here before we kind of go into our example, because blood type can get a little messy. So in codominance, it says that we don't have a recessive. Both are expressed at the same time. I have a little cute, look how cute he is, a little cute blue bird on the left side, and I have a cute little white bird on the right side. Now let's say these two mate and they breed offspring. According to codominance, when these two mate and breed offspring, we are going to have an offspring that has both the blue and white trait in its genotype, which is why we get a blue and a white bird. We don't have just a blend of the two. It's not like we have a lighter blue bird. We can see both white and blue in this bird's phenotype. This idea of codominance is what explains some of your blood types. If you don't know your blood type, I highly recommend that you learn it or you at least know it. But if a parent has a blood type of A and the other parent has a blood type of B, their offspring have a pretty good chance of having the blood type AB. Rather than it just being A blood or B blood, you will have both A and B blood. All right, let's use our example from the previous slide. One bird was homozygous dominant for blue, the other bird was homozygous recessive for white. And let's figure out their chances of having a blue and white bird. We know that these genotypes can show codominance, or when both traits are shown at once. They are not a blend, rather they are kind of like a spotted, we can see both of them at the same time. So let me fill out my Punnett square here. I drop down parent 1's genotype of the big B. I move over parent 2 genotype for the little b, and I end up with a Punnett square that shows me that I have a 100% chance of having a blue and white bird. Not a light blue bird, but a bird that has both blue and white on it. Codominance is when both alleles are expressed at the same time independently. So we've been using flower color as an example, and sometimes in flowers we see codominance. The flower on the right has both red and white at the same time. Codominance is not a blend. It's both colors seen at the same time, right? This explains, this idea of codominance explains spotted cows, right? Depending on your kind of view of this, is a cow black with white spots or white with black spots? Well, it doesn't matter because codominance says that both alleles are expressed at the same time, just separate of each other. A black and white cow doesn't produce a gray cow. We get a black and white spotted cow as a result. So I want to make this very, very clear because it's going to be very easy for you to confuse the two. Codominance is when both alleles are expressed at the same time independently of each other. You can see both black and white in that cow. Incomplete dominance is when the dominant and recessive allele is expressed as a blend. So red and white make a pink flower. Another example of this is blood type. These are my possibilities for my blood type. I can either have AO if an A and O blood cell are blended together. I can have BO when a B and an O red blood cell are blended together. And I can get an AB blood cell when an A and a B blood cell come in contact with each other. It doesn't matter. Codominance means that both alleles are expressed at the same time. Incomplete dominance means that I have a chestnut horse at the top who has dark brown coloring. I have a cremello horse that has white coloring. And when those two come together, I get a palomino horse, or some sort of intermediate or middle color between these two horses. So codominance is independent of each other. Incomplete dominance is a blend. Think of codominance as your coworker, someone who does the same thing as you at the same time, just they aren't a part of you. You are separate of each other, but you are both working to get that done. All right, one more visual to hopefully kind of bring this all home for you. Incomplete dominance and codominance are different. Let's start with a red and a white flower in our parent generation here. Let's say that I breed these two flowers together. I can either have incomplete dominance happen and get a pink flower, or a blend of both phenotypes. Or I can have codominance happen where I see both red and white as a result. So incomplete dominance is a blend. Codominance is when we can see both phenotypes at the same time. So some of you may be wondering why this codominance and incomplete dominance happens in the first place. Well, what we found out in the last 100 years of studying genetics is that Mendel's idea of genetics was really simple, still very significant, but definitely very simple. And now, after studying them for so long, we now know that genetics is not simple at all. In fact, we've discovered these things called epigenetics, which give us different types of alleles. And now this brings us beyond just dominant and recessive traits. We can have different types of recessive alleles. We can have different types of dominant alleles. And this explains our incomplete dominance and codominance. Let's look at rabbits for an example. I have either a black rabbit, a chinchilla rabbit, a Himalayan rabbit, or an albino rabbit. And it all has to do with their genotype. Take a look at their genotype. We're looking at the letter C. If I have two homozygous dominant Cs, I get a black rabbit. If I have two homozygous recessive Cs, I get an albino rabbit. However, it's these little superscripts that make a difference between the two. I can have a homozygous recessive rabbit that has these little CH superscripts up on top, and that's going to give me a gray rabbit, or a blend of the two, or incomplete dominance. I can have a homozygous recessive rabbit that has the superscript or exponent of H, and I can get a Himalayan rabbit that has some white and black, or both alleles being expressed at the same time, otherwise known as codominance. And so when we have these different multiple alleles working at the same time, this is how we can get incomplete dominance and codominance happening. Important takeaways from today. Incomplete dominance is a blend of traits. Codominance is both traits being expressed at the same time. So ladies and gentlemen, if you are confused about any of the information that we just kind of went over in this lesson, I have put multiple links in the description of this assignment to kind of help you. Definitely recommend watching through the Amoeba Sister video. They do a really good job of breaking each of these ideas down and explaining them. If you need any help, questions, you have comments, concerns, please make sure to message me through a Remind or Google Classroom, and I'd be happy to help. But I hope that all of you have a great day, you are safe, and you make good choices.